11. So this last unit, Unit 3, is all about command and control. You know, when we started with Unit 1, we talked about the different levels of organization of the body. So we talked about chemicals and molecules and cells and tissues. And then we got to our first organ system, which was the skin. And then in that middle unit, it was all muscles and bones. So Unit 2 was support and movement. So the muscles and bones and how they move. This third unit is command and control. In other words, how is the muscular system and skeletal system controlled by the nervous system? So so we'll start off talking about neural tissue, like how nerves work, kind of like we did with muscle tissue. We talked about how muscle works, and then we started talking about specific muscles. We're going to do the same thing with the nervous system. So overall, schematically, the nervous system follows this pattern. We have receptors, and receptors create information based on the environment around us or based on changes inside, things like blood pressure, heart rate, that kind of thing. So receptors create a stimuli or a response in response to something. That stimuli from the receptors then has to make its way into the central nervous system. So we have three main kinds of receptors. We have what we call the somatic sensory receptors. These are all the sensations from the skin. So this is touch, pain, temperature, vibration, position. All of those things come from the skin. So we call those somatic senses. We have the visceral senses. Viscera means organ. So while we're not consciously aware of it, at least not most of the time, our organs are providing information to the central nervous system too. The brain can't make decisions about things if it doesn't have information to work with. So we have visceral receptors, and then we have what we call the special senses. And we'll spend a whole chapter talking about those. Those are the five senses that you learned about in grade school. So smell, taste, vision, hearing. We would add balance into there because balance is a separate sense from hearing, even though we think of it together. So we'll cover those in a later chapter. All right. So all of that information from all of these receptors goes into the central nervous system by way of the peripheral nervous system. So we have our first division. The central nervous system is the brain and spinal cord. Period. That's it. All of the other parts of the nervous system are peripheral nervous system components. So these are the nerves, that bring information into the brain and out of the brain. These are the ganglia, or areas of cell bodies in the peripheral nervous system. And we'll talk in subsequent chapters much more about the peripheral nervous system, and then we'll talk about the CNS too. But for our overview, the sensation from the receptors comes in through the sensory division of the peripheral nervous system. That's sort of the input system. Stuff going up into the brain is the sensory division. And then inside the central nervous system, we have information processing. So that stimuli, that information from the receptor, the brain is going to do something with it. It's going to create an action in response to it. It's going to create memory. It's going to create emotion. All of those different things. So the sensation enters the CNS, and there it's processed somehow. It's changed. And then what comes out of the brain then is an effect. And that might be on skeletal muscle, to cause a movement. It might be to affect a gland. So the coming out of the central nervous system is the motor division of the peripheral nervous system. So we have a sensory division and a motor division. Sensory division carries information into the brain. Motor division carries it back out. Now, the words we often use to describe that are afferent and efferent. So afferent is going in, and efferent is going out. So one way to remember the difference, efferents make effects. See how those two words are very similar? So the efferent division of the peripheral nervous system is the effector. In other words, it's signals coming from the brain out to some target organ that are going to make something happen. So afferent is sensory information coming in. Efferent is effect information going back out. So here again, we have another division. We divide the motor division into the somatic nervous system. This is muscle control, basically, skeletal muscle control. So all of the different motions that muscles can make, they're under the guidance of the somatic nervous system. So when we looked at that neuromuscular junction, where the nerve connects with the muscle, that nerve was part of the somatic nervous system. So we'll come back to that in a later chapter. And then the other branch of the motor division is the autonomic nervous system. And we'll spend a lot of time talking about it because it has a lot of importance to healthcare. You know, to how do we take care of patients often has at least a little to do with the autonomic nervous system. So we'll talk a lot about that later. These are the automatic things like heart rate, blood pressure, control of digestion, control of urine formation. All of those things are under the control of the ANS, even though we're not consciously aware of it. So the two divisions of the efferent part of the peripheral nerves are the somatic nervous system and the autonomics. And then finally, we complete our U loop here, down here at the effectors. Essentially, you know, nervous system physiology in a nutshell is stimuli creates response. So stimuli comes in through receptors, and the response comes out through the efferent system. And it's either going to affect skeletal muscle in the case of the somatic nervous system, or some part of the body from the autonomic nervous system. So the digestive tract, the heart, the lungs, the diaphragm, those things. So that's sort of an overview of what we're going to be talking about now for the next several weeks. Yeah.
So would that be at like number 2, where it's actually going into the central nervous system?
Yes, afferent. This would be the afferent.
Okay, and then the afferent would be 4.
And then this is efferent.
Okay.
The whole thing, yeah. All right. So big, long overview. Okay, so that's the giant picture. Now we're going to zoom way in, and we're going to look at nerves and how nerves work. And then we'll zoom back out and look at those different parts of the nervous system in subsequent chapters. All right. So some of this should be review. You've all seen a neuron before in previous classes. Important to note that the scale is different. Neurons are much larger than typical cells in the body. So, you know, like if you look at an epithelial cell under the microscope and a nerve cell under the microscope, one is going to be much, much bigger than the other. So neurons are huge. In addition, because of their evolved design, the axons can be very, very long. So, for example, you have a spinal nerve that goes from the base of your spinal cord all the way to your big toe, let's say. So that's a span of about 3 feet in most people, and that's a single cell. So nerves play by different rules. You know, we don't think of a cell being 3 feet long, but for nerves, they can be like that. You know, the cell body's at one end, and there's a 3-foot axon in between. All right. So the cell body of the neuron, this is where all the work gets done. So this is where energy in the form of ATP is produced. Neurons are very energy intensive. You know, just like muscle has a ton of mitochondria because of all of its ATP use, neurons do too. So they have lots and lots of mitochondria that produce ATP for the entire cell. The other thing that nerves have to be able to do is produce neurotransmitters. You know, the primary function of the nerve is to release neurotransmitters in response to a signal, which we'll talk about more next time. So in order to produce those neurotransmitters, you got to have the machinery to do that. Those are ribosomes. Remember, ribosomes produce proteins. Most neurotransmitters are proteins, or at least can be made in the same way that proteins are made. So we have these Nissl bodies, they're called, Nissl. And these are clusters of ribosomes that are both free and bound to rough endoplasmic reticulum. So when we look at nerves, we see these dark spots in the cytoplasm, and that is where neurotransmitters are being made. All right. And then what we don't see in neurons is a centriole. And I know you may not even remember what a centriole is because it's been a while. But a centriole is an organelle that allows two cells to divide. Neurons don't have those because neurons don't divide. They're a unique cell in the body. Well, not unique, but most cells regenerate. They divide to replace themselves. Neurons don't. They can't divide. And that isn't a bad thing. A lot of times we think, well, it's too bad that nerves don't regenerate. We could grow damaged brain back. But if you think about the nervous system in terms of what it does, if nerves divided, you'd have a very complicated problem because, they don't divide. So the brain has a kind of series of connections from cortex to spinal cord to arm, let's say. If nerves divided, you'd have now two paths to the same part of the body, and things would get confusing. So one of the reasons nerves don't divide evolutionarily is if they did, it would confuse the system because the way the nervous system works is it's all point to point. And as long as that line doesn't change, the nervous system works. So it's not necessarily a bad thing that they don't divide, but it is important to understand that nerves don't grow back, basically. Okay, so the rest of the anatomy, the dendrites are these branching or tree-looking things that come off the cell body. The dendrites are sort of the input branch of the neuron. They're going to collect information from other neurons, and then in the cell body, particularly in the axon hillock, the decision will be made as to whether to send a signal or not. So the dendrites are sort of the receiver of the neuron. The most pronounced feature is the axon, and that's this big, long extension that goes from cell body all the way to the end where the synapses are. The first part of the axon is called the axon hillock, or the first segment, the primary segment. And we're going to talk a lot about this area, so you might want to highlight that in your notes. The axon hillock is an important region in the neuron because that's where the determination to fire or not fire happens, is at the axon hillock. All right. At the end of the long axon, we get some branching. We call those telodendria. And so those are the little finger-like extensions that emerge from the single axon. And then at the end of each telodendria are the synaptic terminals, or the synaptic knobs, we often call them, because they have a little widened area at the very end. So some neuron anatomy. If we zoom way into the synapse, what we can see here, here's a telodendria. So to orient you, we're here. We're going to zoom in to this area right there. And so we see the telodendria, the widened synaptic knob. And then on the inside, we find a mitochondria because this region needs energy. And remember, the mitochondria for the cell or most of the mitochondria are way up there in that cell body, which might be as far as 3 feet away. So good to have a mitochondria around locally to provide energy. All right. Inside the synapse, we have all these little green vesicles. Synaptic vesicles are full of neurotransmitter. And we talked a little bit about this in our muscle chapter when we looked at the neuromuscular junction. So these vesicles are full, and then when the signal arrives down the axon, they're released into the synapse and create a change in the subsequent cell, so in the next cell in the chain, so to speak. Now, important to note, these neurotransmitters, their site of operation is at the synapse, but they're produced in the cell body. So if they're made here, but are needed down here, well, there has to be a mechanism for moving them down this big, long axon. And that's called axoplasmic transport. And it's important because without neurotransmitters, the nerve can't do anything. But the neurotransmitters have to be transported from their site of creation all the way to their site of use. And because nerve cells, particularly in the peripheral nervous system, can be so long, that's a big undertaking. So there's a lot of energy that the neuron uses just to move neurotransmitter down to where it's needed. All right, so that's axoplasmic transport. We often will talk about presynaptic and postsynaptic cells. So the presynaptic cell, we're using the synapse as a kind of line. So the cell in front of the synapse is the presynaptic cell. The cell after the synapse is the postsynaptic cell. And we're going to use this terminology when we talk about how nerves actually communicate with each other. So it's good to just hit those vocabulary words initially. So presynaptic is what is the cell releasing neurotransmitter, right? And postsynaptic is the cell receiving neurotransmitter. All right. Let's see. So there are multiple types of synapses in the body. Three main ones, though. The first is nerve to nerve. So neuron to neuron. That's a synapse. The second we've already seen, and that's the neuromuscular junction. Neuron to muscle. Acetylcholine is released, and that whole cascade of events happens that causes that muscle to contract. One we haven't really talked about is the neuroglandular synapse. This is where a neuron synapses with epithelial cells in a gland. So the adrenal gland, for example, you're watching a scary movie on TV and you get frightened and you feel your heart rate go up. Well, the reason for that is your brain has sent a signal to the adrenal gland to release adrenaline. So that's an example of a neuroglandular synapse where the brain has made something chemically happen. And we'll see more of those when we talk about the autonomic nervous system, as well as in the next semester, you'll see quite a few of those. All right, so three main types of synapses. And then there are more than one kind of neuron. Just like, we talk about the cell, like there's a typical cell in the body that we're talking about, even though there isn't. In many ways, we do the same thing with neurons. We've all seen the picture of big cell body, long axon. The truth is, not all neurons have that body appearance. Like they don't all have the same anatomy. So there are a number of different types of neurons based on their structure. So one is the anaxonic. This is the mystery neuron. There are a ton of these in the brain, but we don't really know how they work or what they do, because they appear to not have an axon. They just have a lot of dendrites and a cell body. So if they have no axon, they can't send any information, so then what's the point of having them there? So we don't really understand what anaxonic neurons do, but they're only found in the brain. So that one's a quick one. Bipolar neurons, that looks like this. They're called bipolar not because they get depressed and crazy, but because they have two poles. They have an up and a down. So their dendrites are very long here, and then the axon is about as long as the dendrites, and then the cell body is in the middle. So we find these in the special senses. So think eyes, ears, taste, smell. And what these neurons do is they carry information from here down to here. Typically, they're sort of like information transmitters or wires. All right, so that's a bipolar. In the unipolar, we find these in the peripheral nervous system. And when you first look, you might think, well, how is the bipolar different than the unipolar? It's all about where the axon is. So for the bipolar, this is all dendrite here, and this is all axon. For the unipolar, this is dendrite, and all of this is axon. Do you see the difference? So in the unipolar neuron, the cell body kind of hangs off to the side of the axon, where in the bipolar, the cell body sits in the middle between dendrite and axon. So we find these are the longest nerves in the body, have this structure, where the cell body is sort of in the middle, which makes sense because remember, the cell body is where all the neurotransmitter and energy is being made. So it's better to have it in the middle so that it can provide equally for this end and for this end. So it's sort of an efficiency thing. And then we have the multipolar neuron is the one we typically see pictures of when you see definition of neuron. You'll see a picture like this. So lots of dendrites around the cell body, and then a long thin axon that ends in a sort of branching pattern. All right. So multipolar neurons are the most common and make up most of the CNS, or the central nervous system. All right. Okay. So if we look at the nervous systems, the central nervous system and the peripheral nervous system, sort of conceptually, this is the kind of thing we get. And I'm not going to go over all of this in class because it's one of those diagrams that I think is better read than being read to you. But it's essentially what we talked about right at the beginning. We have different kinds of receptors that create signals into the nervous system based on changes. So interoreceptors, intero means inside. So interoreceptors monitor things inside the body, the digestive tract, the respiratory tract, the cardiovascular system. So changes or effects from those systems are detected by interoreceptors. So for example, in your aorta, you have a receptor for blood pressure. You've been laying down and you stand up real fast. Well, that receptor senses the drop in your blood pressure, sends a signal to the brain. The brain responds by increasing your heart rate, and your blood pressure stabilizes. So that's an example of an interoreceptor. Nothing in the outside world has changed, but your blood pressure changed because your body position changed. So that's an interoreceptor. An exteroreceptor is just the opposite. This is going to bring information in from the outside world. So this is touch, taste, smell, hearing, vision, the typical senses that we think of. And very often these exteroreceptors can reach conscious awareness. In other words, you can know that stimulus just happened. Consciously you can. So those are the exteroreceptors. And then the proprioceptors, they monitor body position. And they have, for as important as they are, they don't get much press, so to speak. They don't have much publicity. In all of our joints and in all of our muscles are several different kinds of sensors that tell the brain where the body is in space. For example, if you close your eyes and somebody moved one of your fingers for you, you could tell that, not just from the touch of their finger on yours, but because the position of your finger had changed. So that's proprioception. It's critical to balance and coordination. If the brain doesn't know where the parts of the body are, it can't make intelligent choices about it. So you can see some of the effects of this. Alcohol in quantity interrupts the way the cerebellum, which is part of the brain, deals with proprioceptive information. So one of the reasons that drunk people fall down is because the proprioception is off. So it shows you how important that those sensors are to how our musculoskeletal system works. All right, not that you should try that experiment ever. Okay, so all of these receptors the information has to get into the central nervous system. So we have sensory ganglia. All right, vocabulary, ganglia. A ganglia is a collection of nerve cell bodies, neuron cell bodies, that's found outside the nervous system, or outside the central nervous system. So when we get to the spinal cord, we'll see that the cell bodies for our sensory nerves are not located in the central nervous system. They're actually located next to it in a sensory ganglia. The axons from these sensory ganglia then send their information into the central nervous system where that information processing occurs. So the way our brains work, how do we turn stimuli into response? Well, in the brain, thousands of neurons can be involved between stimuli and response. So, for example, if an instructor hands you a term paper, you need to complete this term paper by the end of next week. So, stimuli, right? The eyes see the black and white on the page. The brain recognizes the words, creates meaning out of those words, and then 2 weeks later you go to sit down and do the term paper, and now you're creating a response to that stimuli, right? But you can imagine that is going to involve huge numbers of neurons, a complex task like writing a term paper. So that's what the brain does best, is very complex tasks that can involve thousands, hundreds of thousands, even millions and billions of neurons. So that's the brain. In the spinal cord, there's still information processing, but it's very simple. So, if you've ever reached for something that was hotter than you thought and you dropped it before you realized it, that's the kind of information processing that the spinal cord does. Rapid, quick, but simple. It's not very smart, but it's effective and fast. All right. So we'll come back to these connections as we get further on into the chapter. Let's see. Let me make sure I covered all these. So afferent fibers, those are going in. The receptor types. Efferent. Somatic just means of the body. Typically, that's skin for a sensation and skeletal muscle for effectors. So the somatic nervous system is out to the body and back in from the body. Viscera, we talked about ganglia, interneurons. Okay. Interneurons, that just means neurons between sensation and effect are interneurons. All right. In nervous tissue, there's more than neurons. Neurons are so specialized for what they do, which is to transmit information very rapidly and to process information in very complex ways. Because neurons are so specialized, they need a lot of help to do the stuff normal cells do very easily, like creating energy, like metabolizing nutrients, like amino acids and glucose and things like that. So when we look in at nervous tissue, we don't just see neurons. So the neurons are here in blue, right? All of these yellow cells are what we call glial cells, neuroglia, G-L-I-A, glia. And these glia are the non-neuron parts of nervous tissue, and they're critical. So we're going to look at some of the different types. All right. So up here in pink, these are ependymal cells. Ependymal cells create cerebrospinal fluid, otherwise called CSF. And when we go to talk about the brain and the spinal cord, we'll talk more about what CSF is and why it's important. But for now, ependymal cells create that cerebrospinal fluid. So they're modified epithelial cells that secrete this substance. All right. The microglia, which are here, I don't think they gave us another example, but the microglia, not surprisingly, are small. That's how they got their name. And they serve the same role. They're phagocytes. So, we've heard about like the macrophages. Macrophages are white blood cells that destroy and attack things that don't belong. The microglia perform that function for nervous tissue. So they clean up debris. They keep everything spick and span, essentially. Anything that doesn't belong, they're going to gobble it up and destroy it. So the microglia are kind of like the cleanup crew of the nervous system. Astrocytes, they make up the majority of the glia by mass. In other words, of all the nervous tissue, a lot of it is astrocytes. And that's these guys right here. They get their name because they look star-like. If you lots of points, So the astrocytes have these processes or feet that reach out to the capillary. So this is a blood capillary right here. So that you can see how their feet sort of wrap around the capillary. And then they also have feet that wrap around the nerves as well. Astrocytes play lots of roles. One is they help to regulate the environment of the neurons. So they can pick things up from the blood and put them in around the neuron to keep the environment around that neuron very stable. Another important thing that the astrocytes do is you see how all these feet together have sort of completely covered this capillary? We call that the blood-brain barrier. All right. Because the nervous system is so sensitive to toxins and to chemicals, in our brains, all of the capillaries are sort of sealed so that nothing can get out into the nervous tissue without passing through these astrocytes. So it helps to protect the neurons from what might be coursing through the blood. Now this comes up clinically because a lot of times, we want to give a patient a medication and have it affect the brain, right? Let's say in psychiatry or in a meningitis, a brain infection. Because of this blood-brain barrier, a lot of the chemicals we can put in the blood will never get into the brain because they can't pass through this barrier. So any medication that affects the brain has to be able to pass through this blood-brain barrier. In other words, it has to be taken up by the astrocytes' feet here and actually given to the neuron to have any effect. So it's why the chemicals that can affect the brain often have to be very simple, because it's only simple substances that can pass very easily through that blood-brain barrier. All right. So that's the astrocytes. Metabolic help and forming the blood-brain barrier, keeping the neurons safe. Then the last kind of glia in the CSF are these guys right here, and they're called oligodendrocytes. And what they do, oligo is many, and dendro is hands or feet. And you can see that here's a cell body, and they have these two feet that kind of wrap around the axon. Here's the nerve, and passing through this tunnel, so to speak, is the axon. So we're going to talk more about myelination in a minute, so we'll see these guys. But so here's the axon, and now you can see that this oligodendrocyte has wrapped itself around that in this broad, flat sheet. So they provide the myelin. So that's all central nervous system glia. So ependymal cells, microglia, astrocytes, and then oligodendrocytes. In the peripheral nervous system, there's glia too. Not as much, but still some that are important. So we have these guys, these little cells that are sort of hovering around the neuron cell body. Those are satellite cells. They play the same role astrocytes do in the CNS. So they help the neuron do the normal things of cellular life, like creating energy, like metabolizing things, creating proteins, stuff like that. So the satellite cells do what the astrocytes do. And then instead of oligodendrocytes wrapping around the axons, we have Schwann cells instead. So Schwann cells, so here's the axon, here's the nucleus, and you can see that the Schwann cell has kind of wrapped itself around there. That's how they work. Now, the difference between Schwann cells and oligodendrocytes, one oligodendrocyte can myelinate more than one axon. Do you see how this one is myelinating 2 different axons and this one is 2? For Schwann cells, they only myelinate 1. So the Schwann cell wraps around it wraps around it in a different way. So here we have multiple axons inside the Schwann cell. So why is myelin important? Why do we have these oligodendrocytes that myelinate in the CNS and Schwann cells that myelinate in the peripheral nervous system? It's because myelination speeds up nerve conduction. And we're going to talk about how that happens, which is called saltatory propagation, next time, because we're not going to get there today. But what is myelin? It's basically fat. It's a lipid-rich substance that insulates the axons from their surrounding environment. And we'll see that axons work electrochemically. So by sealing the axons inside this fatty covering, it means that the electricity can move faster. Kind of like insulation on a wire is the easiest way to think about it. Why do we have insulation on the wires we use? So the electricity doesn't leak out, right? Same thing with the myelin. It keeps the electricity from leaking out, and that way it can move faster. So CNS, oligodendrocytes, peripheral nervous system, Schwann cells. All right. So even though nerves don't divide, so they can't replace themselves, they can regenerate to some extent. So the classic example is a peripheral nervous system cell that's had its axon severed. So let's say you had a very deep cut, and one of the nerves that goes to your hand was cut. Well, what's happened is that injury is like right here. So the cell body is fine, for the nerve going to your index finger, because it's all the way back in your spinal cord. It hasn't gotten anywhere near the injury. It's the axon that's been damaged. So so cell body here, axon. Here's the site of injury. Anything distal to the site of injury is going to die. So this axon is going to disappear because it doesn't have the support of the cell body. Remember, with the cell body is where all the machinery is for making energy, for making neurotransmitter, cell membrane, and all that. So if you cut off a part of axon, this is going to die because there isn't anything there to take care of it. Now, the myelin is going to stay. These cells distal to the injury are fine. These cells proximal to the injury are fine. It's only this area that's been damaged. So a thing that can happen is this Schwann cell will repair itself, and if it repairs itself correctly, the axon may be able to grow back to where it was originally by sort of following the tunnel of the Schwann cells that are present there. So we call this Wallerian regeneration, because Dr. Wallerian is the one who discovered it. Because originally we thought that nerves never grew back. But there are instances where people have lost nervous function, but then it has returned. And the reason for that is the axon can grow back. This cell is still very much alive. And it may be very healthy, except for its axon has been cut. So slowly, the Schwann cells reform, and then the axon grows along the length. And if, this is the sketchy part, in order to get function back, it has to go back to where it originally was. And that is what doesn't always happen. Because as this axon is growing, it may wander off in a different direction. It may get stuck up against something where it can't go over or under it. So Wallerian regeneration fails when the axon doesn't get to where it was originally. It is an example, though, of how nerves can grow back. Yeah.
I know that in one of my other classes we were talking about, because originally I had thought that you were only born with a certain number of nerves,
Mm-hmm.
but I guess there's recent research that says that you can get more.
Yeah. Well, you have the brain continues to grow neurons in childhood. Yes. So the number of neurons you're born with is not the number you're going to end up with. Now, what people think is that at some point, around puberty, nerves don't make more of themselves anymore. Does that kind of make sense?
Okay.
So yeah, that's an old myth. You're not born with the same number of neurons you'll end up with.
Okay.
Children, one of the reasons why, children develop so amazingly is because their brains aren't done developing yet. They're still growing new brain while they're learning. So, yeah. All right. Okay, so let's do some questions to wake everybody up. The synaptic vesicles contain neurotransmitters and are present where? Oh, I got to send you the question. Hold on. Okay. There we go. All right, jump in there if you haven't. All right, synaptic vesicles we actually encountered in our muscle chapter too. And these are found in the very end at the synaptic knob. So they're present in the presynaptic terminals. Also, you could call those the synaptic knobs. You could call that the end of the telodendria. But presynaptic, remember I wanted you to learn those words, presynaptic and postsynaptic. So the presynaptic membrane is where the neurotransmitters are released, and then the postsynaptic membrane is where they're going to have their effect. Because you'll see why that's important next time when we start talking about communication. All right, neuron that conducts pain sensation to the central nervous system would be which kind of neuron? All right, jump in there. All right, good. Almost all of you got that. B, sensory or afferent neuron. Everybody hates these words, right? Because they're foreign. You know, we don't use words like this. They sound practically the same, but they have opposite meanings, right? But unfortunately, they're here to stay, particularly in the nervous system. And those of you that are going to go on to like OT, for example, these words are going to come with you. So afferent is the sensory branch, information coming up and into the CNS. Efferent is the effector branch. So a pain sensation is coming into the system. So that would be an afferent branch. All right. One more, a neuroglia question. A neuroglial cell that is a major component of the blood-brain barrier is which of those? I think that is everybody. Okay, so correct answer A, astrocyte. Astrocytes are responsible for creating the blood-brain barrier, and they also assist the neurons in their metabolic work, so in the stuff they need to stay alive. Microglial cells are cleanup. They're phagocytes, so they gobble up and destroy anything that doesn't belong. Oligodendrocytes create myelin, so they myelinate axons in the central nervous system, and then ependymal cells produce CSF. All right. Okay, so for next time, the next two lectures are some of the hardest material in the ANP1. Just like that muscle stuff was complicated with all of the different steps, we're getting to some of that stuff in this chapter too. So even if you're a person who doesn't read the book in advance, please do this time. Look at the next time we're going to talk about resting potential and graded potential, and we may get to action potential. But have looked at this because it gets complicated. All right? So I'll see you all on Friday.
So with the, for example, in Alzheimer's, because that's excess beta protein, isn't it?
Yeah.
Right? So with
Well, the truth is, there is this excess protein, but we don't know if it has anything to do with what Alzheimer's actually does.
Okay.
It's weird. We know that there are these plaques, but we don't know what causes Alzheimer's disease.
Okay.
They're associated.
So would that buildup of protein with the microglia supposed to be
Yes, the
taking care of that?
microglia should be taking care of that, yes. But the protein plaques may escape notice is the thing. Because they can't see, so these phagocytes, they can only destroy something they recognize as foreign.
Okay, and there is beta, like your brain naturally produces.
Yes.
This. It's just like cancer, where like it happens, but without it
Correct.
being.
Well, and it's just a funny thing, in Alzheimer, we don't know why, but the brain atrophies. In other words, it literally shrinks.
Mhm.
Its connections start to disappear, and we don't know what the connection is with this beta protein. We don't know why we see beta protein in Alzheimer patients. Because it isn't even diagnostic. Like if you did a brain biopsy, you say you think somebody might have Alzheimer, and you did a brain biopsy and there were no plaques, it doesn't tell you they don't have it.
Mhm.
And if there were plaques, it doesn't tell you they do.
Okay.
So it's a bit unknown still.
Yeah.
So it's a diagnosis of exclusion. You have to rule everything else out.
Mhm.
Oh, I didn't stop.